Okay, hello everybody. This is going to be a presentation on our paper, Neuro-Evolutionary Transfer Learning through Structural Adaptation. My name is Josh Karns, and I'm a first-year PhD student at RIT. Co-authored is Abdelrahman, who we just heard, another grad student, Zimeng, and three professors from RIT, Professor Daniel Krutz, Professor Alex Ororbia, and Professor Travis Desell. Okay, so first, since this paper is about transfer learning, I just want to add a brief word about what transfer learning is and the general idea in the context of artificial neural networks is that if you given an artificial neural network that is trained on a particular data set, it may be a useful starting place for a similar but distinct problem, right? So it will allow you to avoid starting from scratch, which can reduce convergence time, give you improved anytime performance, and give you better absolute performance. Next, I just want to mention what neuroevolution is briefly. So applying evolutionary algorithms to neural networks, meaning using evolutionary algorithms to find optimal neural network architectures and their associated weights. And like with most evolutionary algorithms, it uses mutation and crossover. Okay, so in the previous presentation, we heard this exam term get thrown around. So exam is a neuroevolutionary algorithm, evolutionary exploration of augmenting memory models, and it generates recurrent neural networks and finds their weights via backpropagation. The weights are pre-initialized from parent neural networks, so we don't lose progress. We don't have to restart the training process every time we get a new recurrent neural network. Yeah, so it evolves recurrent neural networks using mutation and crossover operations with both node and edge level mutations. And it's an asynchronous island-based evolution strategy. The islands are separate populations of genomes that are less likely to perform crossover with each other, which kind of helps us explore the search space a bit more. And yeah, crossover occurs within and between islands, but less frequently between islands. Next, since we're using recurrent neural networks, they're commonly used for classification type problems, but I just want to mention that recurrent neural networks can be used for time series data prediction as well, which means we're going to be doing something like mean squared error or optimizing for mean squared error or mean absolute error. And in this paper, we use mean squared error. And yeah, this is the data sets we're using are going to give us regression problems, which are quite difficult. Next, I want to talk about roughly how exam works from like a high level. So we have a master process which keeps track of all of the genomes. That is, it keeps track of all n of our islands and all m members on each island. And then we have a pool of workers, and the workers basically, they will request a recurrent neural network from our master process. The master process is going to generate a recurrent neural network through mutation or crossover. And it's going to send it over to the worker process. And the worker process is going to perform backpropagation on it for a couple of epochs. And then it's going to calculate the mean squared error on a testing data set that is not the training data set, and send that back over to the master process. And that mean squared error is going to be our fitness, and it's what we use to figure out if we should insert a given genome into the population. If it's better than the worst thing in the island that it belongs to, it's going to be inserted. Otherwise, it gets tossed, and we rinse and repeat until we get something we like. So going over the operations that exam has briefly, Our crossover operation here, there's always going to be a parent with a better fitness and a worse fitness. In our crossover operation, everything that's in the better parent is going to be in the child network shown on the right here. And the things that are in the worst parent will be in the child network with a probability of 0.5. Next, I'm going to go over some of the mutations we have. So we have a split edge mutation, which will choose a random edge, disable it, and then insert a new node that goes from the original input to our new node, or it'll add a new node, add an edge from our original input to our new node, and then add an edge from our new node to the original output. And this node can also be just a normal, like a simple cell, or one of several, memory cells. Okay. We also have an add edge mutation here, which will just add a random feed forward edge between two random nodes. Next, we have a disable edge operation, which will select a random edge that is enabled and disable it. Next, we have a enable edge operation, which will find a random disabled edge and enable it. We have an add recurrent edge operation, which will select a random, source node, and then a random destination node, which is just the only restriction on the destination node is that it's not an input node. And it'll create a recurrent edge between those two with a time lag between 1 and 10. Next, we have an add node operation, which will choose a random depth, and it will use statistics about the connectivity in the existing network to determine how many nodes or how many incoming and outgoing edges it should have. And those are all determined randomly. Then we have a split node operation, which will select a random node, disable it, disable all of the incoming and outgoing and recurrent edges. And then it will create two new nodes, and it'll split those incoming and outgoing edges between the two new nodes equally. Each of them is going to get half, but if there's only one incoming edge in the original node, they'll both have it because you can't split an edge in half. So each is going to have at least one incoming and one outgoing edge. And then we have a merge node operation, which is going to select two nodes, disable all their incoming and outgoing edges, and then create a new node. And it's just going to be the union of those two in terms of their incoming and outgoing edges. Okay. Then we have a disable node operation, which just disables a node and all of its incoming and outgoing edges. And finally, we have an enable node operation, which will find a disabled node and enable it along with all of its incoming and outgoing edges. Okay. So talking about this adaptive structure transfer learning thing. So normally exam will begin with a minimal neural network that is given the set of inputs and the set of outputs. It's a network that's just well connected, all of the inputs to all of the outputs, no recurrent edges, no nothing other than the simple feed forward connections. So rather than do this, if we want to do some sort of transfer learning, we can use a network trained on a similar data set. And even if this data set doesn't line up with, even if the source data set doesn't line up with our target data set, we can remove the inputs and outputs that don't exist in the new data set and add any new inputs and outputs. And we can use the neuro-evolutionary process offered by exam to sort of fill in the knowledge gaps and take advantage of any new inputs. And to our knowledge, this is the first work to use neuro-evolution for transfer learning and to use a transfer learning strategy of structural adaptation that is removing inputs and outputs in addition to adding inputs and outputs. Okay, so the process for preparing the source network is not that bad. Given our, the neural network trained on our source data set here on the left, our target data set in this case doesn't have output 1, so we have to remove output 1. So we get rid of it and all of its incoming edges. Next, we have to add our new outputs. So we add output 4, and the way we connect it into the network is just connect all of the inputs to this new output 4. And we will repeat that for however many outputs we have to add, but in this case, it's just 1. And then in this case, our source data set has an input 2, and our destination or our target data set doesn't have this input 2. So we're going to get rid of that node and all of its outgoing edges. So we got rid of it. And then we have to add a new input, input 4 in this case. And then we connect input 4 to all of the output edges. And finally, there's sort of some dangling nodes there. That is nodes where there's no path from any input to any output that includes that node or edge. They're highlighted in red here. So those are all nodes and edges that have no impact on the output of the network. So we just get rid of those. Okay, so that's the process. It's not too bad. Going over our data sets a little bit. I mentioned exam evolves recurrent neural networks. So our data sets are all time series data sets. One of them we just saw in the previous presentation from a coal-fired power plant. It's per minute readings of sensors from the coal plant's burners. And our source data set had 12 parameters. And our target data set had 8 additional parameters from installing a new sensor, which gave us a total of 20. And then we have a second data set, an aviation data set, which has per second readings of aircraft sensors during flights. And we have 3 different aircraft, each with a different set of sensors, and each aircraft is fairly different from one another. Okay, going a bit more into the coal data set, the networks were trained to predict the main flame intensity parameter. And both the source and target data sets had 4 files. Each file represented about 24 and a half hours of per minute sensor readings. And they contain those 12 parameters, but they roughly have to do with, fuel conditions, airflow, and the temperature. And then these 8 additional parameters offered by the target data set are a consequence of installing a new full stream element analyzer in the coal plant, which gives us some information about the content of the coal in terms of its elemental content and a few other things. So the experiments we ran on the coal data set, we generated and trained 2,000 genomes on the source data set, that is the data set with the 12 input parameters. We used the best genome from this process as the seed for our target data set. And then we perform our adaptive structure process and begin training for 2,000 genomes on the target data set. And we repeat this process 10 times, and we compared our results with generating 2,000 genomes on the target data set without using a seed genome, just using that minimal network where all 20 inputs are connected to the one output to begin with. And just to give you, briefly to give you an idea of what this flame intensity parameter looks like, in blue is the ground truth actual flame intensity. And we have a prediction kind of on top of it, which makes it hard to see, but it is sort of clear that it's pretty noisy. It's not particularly seasonal, which just can make it a fairly difficult regression problem. And here is this is our results basically. The y-axis here is mean squared error, and we can see that the the transfer learning group, the one in orange, it did perform better. It trained a bit faster, and it achieved better performance overall. The shaded region, the top of that shaded region is the worst performing run out of all 10 runs we did, and the bottom of a given shaded region is the best. So we can see that the best performing one in the transfer learning group is significantly better than the non-transfer learning group. But yeah, no, the benefits here are fairly clear. Okay, so going a bit more into our aviation data set now. We have data collected from 3 different aircraft. There's 12 flight files per aircraft, 2 of which got used as a testing data set, and the other 10 are used as a training data set. Each flight is between 1 and 3 hours, and it's per second sensor readings. And each aircraft data set was used as both a source and a target data set. And so we have these 3 aircraft, the Cessna 172 Skyhawk, which I'm going to call a C-172, Piper PA-28 Cherokee, which I'm going to call a PA-28, and a Piper PA-44 Seminole, which I'm going to call a PA-44. The PA-44 is significantly different from the PA-28 and C-172 because it has 2 engines, which means it has a lot more, sensor readings or input parameters. And yeah, the PA-28 and C-172 are both single engine aircraft, but they have different wing designs, which makes for some notable differences. And they also have a different set of sensors. All 3 of the aircraft have these 18 parameters, which just kind of generally it's about the location and the environment of the aircraft. So each aircraft has a unique set of additional parameters, and most of them are related to the engine. And the networks were trained to predict all of the parameters that have the term exhaust gas in them, which means the PA-28, the networks trained to predict the PA-28 had 4 outputs. The ones trained to predict for the PA-44 had 8 outputs. And the C-172 had 4 outputs as well, just like the PA-28. And the rest of the parameters are just kind of various engine-related stuff. So the experiments we ran, we generated and trained 4,000 genomes for each aircraft, and just starting with the minimal connected network. And we used each one of, or we would use this as the source genomes and transfer to the other 2 aircraft, which gives us 6 experimental groups. And I guess it's just worth noting that anything that sort of involves the PA-44, the 2-engine aircraft, has like the most inputs and outputs removed. Rather, we had to sort of, our adaptive structure process was the most involved for anything that had the PA-44 data set. Okay, so transferring, these are results transferring to the PA-28. We see that transferring from both the C-172 and PA-44 seemed to perform better, although with the PA-44, it took some time for it to kind of actually start doing any better. But it is fairly notable. So transferring to the C-172, transferring from the PA-44 to the C-172 didn't seem to do particularly well at all. But transferring from the PA-28 to the C-172, and these are both single engine aircraft, seem to perform quite well. And the last group is transferring from the single engine aircrafts to the dual engine aircraft, and it seems like that did not work particularly well at all. But it kind of makes sense given the significant differences between the 2 data sets. And just some brief information about the neural networks we actually generated for each one of these groups. So compared to groups involved with the PA44 in any way that is either transferred from or transferred to, the other neural network groups sort of, they just have fewer nodes and edges and recurrent edges, but that makes sense because the PA44 data set had a lot more inputs and outputs, and there's not really much else of interest here. So the discussion, transfer learning using NeuroEvolution can speed up the training process for time series data forecasting, and it can also improve our absolute performance. And there's a lot of room for future research using more sophisticated strategies for removing, adding inputs or outputs. And we have an upcoming Gecko paper about that, and incorporating domain-specific knowledge into the transfer process, just to name a few. As far as acknowledgements, our research is funded by the U.S. Department of Energy and the Federal Aviation Administration. And that's all I got. Thanks for listening. And if you have anything you want to email me about, you can do that. And I can take a few questions maybe.
Hi, Josh. Thanks very much for your presentation. We have time for questions. So please go ahead. Okay, we have a question from Jean Pryor. Maybe you can read it out loud so you get that on tape.
I have to stop screen sharing so I can see the question.
Yeah, probably.
Okay, here. Oh, I don't see the question. Could you read it for me?
So, Jean, maybe you can read it.
Or if you just copy and paste it.
Oh,
Yeah.
sorry. Sorry, I was muted. I was muted on my microphone. Sorry. Thank you for the presentation. I have a couple of questions about this work. Mainly concerned about the initialization. When you say you generate around 200 genomes, what is? Can you tell me about a little about that process of generating? How do you prevent from them to just blow up in structure or in terms of weights, or how do you control it?
Okay, so what is it? We have that neuro-evolutionary process. So when I say 2,000 genomes, I don't mean we generate 2,000. We generate 2,000 using that neuro-evolutionary process. So we don't really control for like the networks blowing up, but if the networks don't perform well, we don't insert them into the population. And we also have an additional operation that I didn't mention. It's a mutation where it doesn't do anything at all. It just clones the network and performs back propagation on it. So that might also contribute to why the networks don't blow up in size.
Okay, that was also one of my follow-up. I was trying to see how if you did some warm-up training for the networks before testing them, but I think you have already answered with that mutation operator, or do you perform?
Every time we generate a neural network via mutation or crossover or just cloning, we train it with back propagation. And each network inherits any applicable weights from its parents as well, which means we don't lose all that progress as well. So that's how that you could consider that to be a warm up.
Okay, thank you.
Any more questions? No more questions, I guess.
So, Penousal.
Yes, Mark.
If there's no more question, I'd like to make a test now. I would like to unmute everybody and then everybody applauds to see how it Zoom reacts.
Okay, sure.
Let's applaud the speakers, for instance, or Penousal or whatever. Okay, I'm doing it now. Please applaud.
So now everybody is unmuted.
Please applaud, everybody. Can we hear everybody applauding?
Yeah.